things on the phone. I just said. Hello, everyone. Welcome to the people in the room. Welcome to the people online. Thank you for the SAIAB community also to join us on this seminar. I think there's not a better way of starting August Women's Month with the seminars that Kerry-Ann is about to give us. Kerry-Ann is a postdoc here at SAIAB. She's working on the physiological tolerance of coastal organisms in South Africa, and she's just been lucky and worked hard to come back from this conference where she attended this conference in Portugal, Sesimbra, and I need my note because it's quite a name for International Symposium on Human Impact of Marine Functional Connectivity. This conference was organized by the Sea Unicorn network, from which we are part of it, and some of the people here at Rhodes also are collaborators. So I don't want to open up more. You're just gonna give it to you.
Sure. Thank you, Francesca.
Enjoy. Thank you.
Great. Okay. Cool. So thanks, Francesca, for that introduction, and good morning, everyone, and good morning online. So I'm very excited to give this talk. There we go. Because it's a talk that I gave earlier this year in May at the International Symposium on Human Impacts on Marine Functional Connectivity. Yes, Francesca, it is quite a mouthful. And it was hosted by the COST Sea Unicorn as well as ICES. And just to give you an introduction of these organizations, COST Sea Unicorn is a large international and interdisciplinary group of scientists and stakeholders who are promoting the concept around an emerging field known as marine functional connectivity. And ICES is a European marine science organization who aim to share scientific knowledge around understanding marine ecosystems and the services they provide for conservation and management. So, as Francesca mentioned, the symposium was held in Portugal in a very small coastal town called Sesimbra. Now, in addition to its beautiful beaches, as you can see over there, Sesimbra is known for its fish. So the characteristics of the water, the different artisanal techniques and that the fishermen use, and the way that the local restaurants prepare these amazing dishes makes this a very unique and special place. So the symposium was held at the Sesimbra Municipal Theatre, and I gave my presentation on the very first day of the symposium, but I was the very last speaker for the day. But considering I was one of 2 South Africans presenting at the symposium, it wasn't hard to keep the audience entertained with our interesting research and beautiful coastlines, and also a great distraction from most of the talks that took place that day, which focused on the Mediterranean Sea. So let's dive into the talk that I gave, shall we? So coastal habitats like your rocky shores and your estuaries are highly productive areas which consist of many different fish and invertebrate species across one or more of their life stages. These coastal habitats are great because their variability in structural complexity, the substrates, as well as the hydrodynamics, improves the habitat quality and connectivity, allowing them to act as great nursery areas for early life stages, which is essential for the maintenance of biodiversity. But there is an effect of coastal urbanization or urbanization, which is replacing these natural coastal habitats with artificial structures such as your sea walls. I don't know if this pointer's working. No, okay. Your sea walls and your jetties and your piers within ports and harbors. So these artificial structures basically fragment this natural habitat by replacing them or partially replacing them, which then influences the biodiversity or changes biodiversity in terms of a reduction of space, as well as preventing organism movement and gene exchange. So they are also responsible for altering flow patterns and wave energy, and they increase sedimentation and can create these areas of low oxygen. In addition to this artificial habitat alteration, we have global warming stresses, such as your rising mean ocean temperatures, your increasing local temperature variability, which is mostly experienced in upwelling events along our coastline, and more severe extreme events like marine heat waves. And this may place additional pressure on these early life stages moving into these coastal urban systems to settle and recruit. And of course, in turn, this will affect their metabolism. It could influence their growth rate and their overall survival. So brachyurans, or true crabs, have got these complex life history strategies. So they mostly comprise of several zooea stages and megalopae, which both depend on both the estuarine and the open sea environments. And these excessively high mortality rates, especially at the zooea and megalopa stage, are highly influenced by chemical, physical, and biological processes. One of the most detrimental physical process would be increasing temperatures, which can affect them. So basically, the changes in these life history traits and the population dynamics are greatly linked to these ecophysiological processes, which is influenced by the environmental conditions. So to show this in a simpler way, an altered habitat with altered environmental conditions can prevent these brachyuran crab larvae from picking up important cues, physical cues, chemical cues that they need in order to settle and metamorphosize. So metamorphosize meaning into the next life stage, predominantly which takes place after they've found a really nice little home. So the aim of this whole project was then to assess how resilient are these larvae in terms of their functional connectivity. And we also wanted to understand what exactly are the impacts of these temperature changes of these larvae within artificial ecosystems. So firstly, we did this by looking at their metabolic performance. So how are they functioning or performing at different various test temperatures that we are testing? And then we wanted to see, a little bit delayed. Okay, we go, sorry. We then wanted to compare the metabolic performance of these larvae to predicted increases in temperature. And we also wanted to assess if artificial structures like those seawalls and jetties would be negatively affecting their ecophysiological responses in terms of their functional connectivity. So the study area that we selected was the Royal Port Alfred Marina, which is found along the Kawi Estuary in Port Alfred. And within the Port Alfred Marina, we had two microhabitats. We chose jetty sites and seawall sites. And at each of these sites, we placed these temperature loggers called environmental loggers, just to give us an indication of the temperature variation that is occurring within the sites or at these sites. But for the purpose of this presentation, I'm just going to be focusing on preliminary results that we got from the Jetty B microhabitat. So the study species, the Brachyuran crabs that we selected were the pea crab, known as Pinothera species, and the crown crab, Hymenosoma operculata. And we were focusing on the zooea and megalopae life stages. So just to show you how tiny these organisms are, at the zooea life stage, they don't reach they can reach up to a maximum of 1 millimeter, whereas the megalopae life stage is approximately 1 millimeter. And then just to give you an indication of how small they are at the adult stage, the Parnotherus crab, pea crab, the name's giving it away, the females reach a maximum length of 13 millimeters, and the males are generally much smaller, being 6 millimeters. And this is because they live in the host of mussels or oysters. So interestingly enough, at the megalope life stage, they pick up these chemicals in the water that the mussels give off, and they find the mussel host and they settle inside the host, and then they develop into that adult stage, basically feeding off on what the mussels feed, the filtering feeding, and also off the mussels. And then the Hymenostoma crab, they are also quite small. They only reach a maximum of about 26 millimeters, and they're a highly abundant species, and they're commonly found within the gut contents of many other fish species, showing how essential they are in the food web. And you often find them in the soft sediments that they're occurring in within whatever habitat they're found. So now that you know the species we were targeting, we collected them using these light traps. Because they're phototactic, they're attracted to the light, and we set these at springtide, and we brought them back to the lab, and we quickly sorted through selecting the stages we want to focus on, and then we measured their routine oxygen consumption rates using closed respirometry. So we have this microplate that has 24 chambers, and we put an individual in each of the wells. And I don't know if you can see it now, but a blue light was flashing earlier, which means that every minute it would measure an oxygen reading. And it would get caught up here on, or processed onto the laptop, and then we would analyze that at a later stage. So moving on to the results, the interesting parts. Before I get into them, I just want you to focus or pay attention to the scales on the y-axis. So you can see that the metabolic rates are a bit higher for the crown crab as opposed to the pea crab. But what we're seeing here is that the megalope for Hymenostoma operculata has got a much higher routine, a significantly higher routine oxygen consumption rate compared to the Zouaille life stage. And you can see that the Zouaille life stage, temperature did not have a significant impact on their routine metabolic rates. Whereas for the pea crab, there was no significant difference in routine metabolic rates between the life stages. But we can see that temperature did have an influence, it did have an influence on the routine metabolic rates, especially for the megalope life stage at temperatures greater than 19 degrees. And also just to go back here, you can see that at 22 degrees and 25 degrees, that's where the megalope seem to be having a much higher routine metabolic rate. So bearing in mind, or keeping this in mind, that the megalope seem to be more at risk in terms of to higher temperatures, if we had to compare these thermal performances to the in-situ temperature data that we collected, we can already see that during the summer months, sorry, I'm using my hands a lot, the temperatures are higher than their optimal temperature, around 19 degrees. And if we had to add 2 degrees Celsius on to this in-situ temperature to illustrate the predicted impacts of a temperature increase in mean ocean temperature, we can see that temperatures are going to actually reach or go beyond the maximum environmental temperature that they are experiencing now, which is 25 degrees. So looking at these preliminary results, we can definitely see that the megalope life stage for both of these species, especially the crown crab, may be more vulnerable to temperatures greater than 22 degrees. And this may be a result of crab larvae becoming increasingly stenothermic as they develop. Now, when I say stenothermic, I'm meaning that they have a very narrow window or narrow thermal window to these temperature changes. So they will probably have higher energy demands. And this in turn may make them more vulnerable to these predicted increases in mean ocean temperature, as well as extreme events like the marine heat waves, which they may already be experiencing. And from data and research, we see that marine heat waves are expected to increase in intensity and frequency within our region. So no, that's not really great. Then also the water temperatures greater than 22 degrees may inhibit molting and settlement of the megalopae. So I'm going to use Hymenosoma, the crown crab, to illustrate this. So at the optimal temperature cues of around 19 degrees, you'll see that the megalopae will have increased swimming activity, and they will move into areas of higher flow rates, and they will generally explore a lot more, looking for a suitable area for them to settle and then metamorphosize or molt into the next life stage. But if these temperatures are higher than what they like 22, 25, and expectedly higher temperatures, we may find that their metabolism is now decreased. So they don't really want to swim around anywhere, and they don't want to generally explore and look for more suitable areas for them to settle. So in this case, metamorphosis and settlement may be delayed. So in terms of the functional connectivity, so with this increase in temperature, or these temperature effects, it may affect their physiology, as I said, and they may be retained within these jetty areas. And this may result in this serving as some sort of a retention area or a sink. And as I said, it will prevent successful recruitment, because in other words, they may be missing an important evolutionary step, which is moving offshore to the open sea to complete their life cycles. In terms of the artificial structures, like the seawalls, which are located along the estuary, and the pier, which is enclosing the jetty area, this may reduce this tidal flushing. So you can see, I try to illustrate this nicely with water movement becoming less and less and less the further it goes up. It may create this stagnant water within the jetty area. And this stagnant water may create these pools of warm water, which may result in the megalopae being exposed to higher temperatures for longer periods, which could lead to potential mortality. So in conclusion, the question I want to ask is, do you think that these crab larvae are tiny titans? Are they able to conquer climate change and temperature increases? From the preliminary results that we've seen, evidence suggests no, because the megalopae are not coping with temperatures greater than 22 degrees, and they may be retained in these jetty sites because of the artificial structures surrounding them and resulting in poor functional connectivity, which could delay settlement and mortality. But on the other side of the spectrum, these species are highly abundant in the jetty area. We are finding them in large numbers, and they are reproducing throughout the year. So could actually these sites be serving as some sort of a hub for biodiversity? So for us to get a clear answer to the question that I've previously posed, we need to conduct more research. And we need to conduct more research in terms of looking at thermal performance across their entire life cycle. So we need to include juveniles, and we need to include adults to see how they are influenced by these effects of urbanization and climate warming. We also need to do more assemblage surveys. So the surveys that we've currently done, we found a lot of megalopae, we found a lot of zoea, but we haven't actually found any adults, and we haven't found any juveniles. So it would be really interesting to go into the field again, maybe go look in some mussels, see if we can find any of those adult crabs. We also need to look at the seawall, compare the data we've gotten from the seawall microhabitat to see what might be happening in these two different artificial microhabitats. And I think importantly, we need to know what are their thermal tolerance limits. What is the maximum temperature that these organisms can actually tolerate? So are they able to tolerate temperatures greater than the environmental temperature that they're experiencing now? What kind of leeway do they have? And we should also include surrounding harbors and natural rocky shores to make this comparison to see if there is a general trend occurring here. So just to end off with my presentation, I thought I'd share a little bit more of the fun part of the conference. After the presentation, I went and celebrated and got this delicious Nutella crepe. And then luckily enough, or fortunately enough, we had a day to explore Sesimbra at the end of the conference. So we did this beautiful hike to the secluded beach. And this was a picture taken from the gorge on top of on top of the mountain there, overlooking the town. And then this was the town area, and we actually stayed up one of these little streets up there. So we're very close to the beach, which was great. But what I really loved about Sesimbra was the fact that every open wall had this beautiful artwork. And as you can see, there's a nice picture of me with an octopus mural there. And then also another fish that they traditionally fish. I can't remember the name now, black scabbard fish, yes. It's a deep water fish that they traditionally have developed these certain techniques to catch them, which is quite interesting. So I hope you enjoyed the talk, and I would just like to acknowledge the NRF and especially the Department of Science Innovation, because they made this trip possible by providing me with a travel grant. And everybody that assisted with the research and the co-authors on this, this paper that's soon to be published. Any questions? Thank you.
Thank you so much for this dive into the trip and the results, the study. Really exciting. You made us live some of your experiences.
Thanks.
Any question from the floor for Kerry-Ann? Yes, there's one here.
I was just wondering, when you mentioned that the jetty sites function as sinks or retention areas, do you think it might be worth looking at population connectivity between sinks, since you might be considering them as hubs of biodiversity and such?
Yeah, definitely. I think that's what I brought on into the end, is that I think it's important we're doing more assemblage surveys and looking at how many are they occurring there, are they occurring in abundance at the seawall. So I think it's important to obviously do that. Thank you.
Would it be like a spatially limited thing, or would you apply it over a large distance, like a large swath of coastal?
Well, I think for the purpose of what we're trying to answer, we're looking at mostly the effects of these artificial habitats. So I think it would be important to include all the microhabitats that could have a significant influence. So like more in terms of seawalls and things like that. But in terms of the natural coastline, I think we would focus on like tidal pools, rocky areas where they were caught previously. So that's probably the area of focus.
Thank you.
Thank you. Any more questions? Yes, Taryn.
Great talk, Kerry. I loved your slides. Beautiful.
Thank you.
Do they actively feed?
They do actively feed, and they are picking up phytoplankton mostly. And I find this is what's so interesting, is that they hanging out in the jetty area, and like there's a lot of food that's probably accumulating there as well. So I think that's probably why they're enjoying this. They like stagnant water, and I think that most of the food kind of like ends up in those enclosed areas anyway.
Yeah, because I'm just trying to think of your curves showing the functional connectivity or functional what is it? You know which curves I'm talking about. So,
yes, the thermal performance.
the megalope was going up. So
Yeah.
if there's what are they called?
Zoea.
Are not actively feeding, then I thought that's why you would probably see this stagnant curve, because they're just cruising around doing whatever they're doing, maybe not actively feeding.
Maybe.
But it's really interesting seeing that flat curve. And then just out of interest question, are they parasites, or do they have a symbiotic relationship with the mussels?
The pea crabs are actually parasites. So what happens is they live inside the mussel, and they're benefiting from both feeding off the mussel at much later life stages, so past the megalope life stage, at the juvenile stage. So they switch diets from feeding off plankton, and then they go more onto like omnivorous food, feeding off the mussel. So interestingly enough, I came across a paper where in New Zealand, they're actually struggling with these pea crabs because they're trying to grow or culture mussels there, and the pea crabs just get in and it that destroys their brood stock, and so they are actually quite pests.
They're
quite a bit
slowly
pests.
just keeping them alive, but slowly killing them.
Yeah, and it also reduces the growth of the mussel as well.
Okay.
So in terms of like profit for culturing these
Yeah.
mussels, you're not going to get the size that they want, basically. So they are parasites. Yeah.
Thank you. I think there's another. I'll take another question from. Okay, this is. Let's take from the online, and I'll come back to you, Amber. Who is asking, are the megalope occurring at the jetty?
So from your results.
So from your result, can you conclude there is a population bottleneck at the megalope stage as a result of the temperature?
So Pule, they are occurring at the jetty, actually in high abundance. The question that we need to kind of now pose is, are they making it past the megalope life stage? So we're seeing that maybe that temperature could be affecting them moving on to the next life stage, but we're not certain. So that's the next step. We need to actually see if they're making it to the adult stage and are they spawning. So there could be a bottleneck. I was scared to say bottleneck or ecological trap because, yeah, it's quite a big concept. I hope I answered your question, Billy.
Let's get one for Amber back to the
Well
floor.
done, that's awesome talk. That's so
Thanks,
interesting.
Amber.
So nice to hear about crabs. And
Yeah.
I love the source of think argument because I mean, it could be a thing, it could also be a source, right?
Yeah.
So, but I wanted to ask you about the mussels. Has there been work done on thermal tolerance of their host?
Yeah, so actually I did do some thermal tolerance work on Perna for my PhD, and they can tolerate quite high temperatures, so they're quite resilient.
Okay, so the host will still be available for them if
Yes,
they tolerate it.
yes.
Okay, that's awesome. And then I thought for future, have you thought about growing them out in the lab so you can do aerobiscope on the different life stages if you can't, instead of collecting them from the wild?
That would be great. I'd love to do that.
And then the last one is that working with such, I mean, no one can actually really, I suppose, understand and appreciate the difficulties working with 1 millimeter with larvae, because it would be awesome to do repeated measures at that young stage, but obviously you can't. There are some more high performance or low performance and the resilience within those populations.
Yeah.
So maybe you could do it if you grow them out at the larger stage. So.
Yeah, that would be a great, thanks, Amber.
That's the adults.
Yeah.
You can't do it for the young.
No, I mean, at that small stage, Seshni was helping me and the Zoea and getting them into those little chambers. It's extremely difficult and it's tedious. You've got to be so precise when you're doing.
But then you could do a Q you can pull a Q and do a Q and look at the adults and see if it's heritable. So those high performance traits are
great.
heritable. So I mean, going forward, that could be a really cool PhD for someone.
Oh, thanks. Thank you, Amber.
Thanks so much, Amber. If do we have more time, Lucky? I just have one question or one potential answer to your. I love the conclusion. Tiny titans, yes or no? Could there be something in the middle? And I mean, at the end of the day, maybe it's a sink, maybe it's a source.
Yeah.
We're not sure if they've been exported, but here there could be a feast waiting to happen for something else. I mean, when we think about that tiny, and someone here in the audience know how hard it is to fill an 80 micron well, but thousands and thousands of this, because we know from Session's work that there's lots of this. Could they make it available for the next level? So how there's very early and very low bottom trophic consumers are, could they enhance something for something else?
Well, I mean, as I mentioned, like the crown crab, they're commonly found in the gut contents of other fish, and they are highly abundant. So I do think that they're essential for the food web dynamics within this habitat. So there could be definitely a maybe, like something else that could be going on. So I think that's why focusing on those research questions at the end to get a clearer idea of what could be going on could answer some of those, could answer that maybe they're just there for food. Yeah.
One last question from Albert. Yes.
Yeah, thanks, Kerry. So what's the distribution range of these crabs? Are they confined to temperate regions, or you find them all along the coast? I'm just curious.
They're actually found. So for the pea crabs, they found all along the coast. They go up to the tropical areas. Lyle found some actually in the mangrove areas, which is quite interesting. So and Hymenosoma, I think they're more temperate. So they don't generally like go higher than they don't go into the tropical or subtropics. So they're more temperate in terms of their distribution. So that could also be a reason as to why they're so influenced by the temperature changes, higher temperatures.
Yeah, because I was just wondering about the cutoff temperature of 22 degrees. If they do also okay in the tropical regions where temperatures might actually be much higher than that.
Yeah, I mean, that's a great observation. Thank you, Albert. And I think that shows quite nicely in the results, because you can see for the pea crab, they're not consuming a lot of oxygen at the higher temperatures, as opposed to the crown crabs, who may be more temperate and they don't like the warm temperatures. So yeah, that's a really good point. Thank you.
Yes.
Thanks so much. As you can see, there's lots of spinning of more ideas. So listening, more students, more funds to do more fun research. So I would like to thank you all, the audience in the room and the audience online. And I'd like one last round of applause for Kerry-Ann. Thank you.
Oh.